Portland, Ohio. Grab my notebook so I can remember what I want to criticize each other about at dinner.
Oh, there's gonna be a lot you can criticize me about.
Please don't criticize me at dinner. I will break down and cry.
Oh, I know. I bugged by it.
No, that's
That's David.
ABRF. It is the same software though.
Oh, you use OBS.
I do use it.
Do you use it with like DaVinci or anything else, or just
So
OBS?
far, it's just been the live streams. I have Caden Live if I decide to edit.
Oh, okay.
Haven't learned how to use Caden Live. I'm aware. The recording is not though.
Yeah. We're gonna say all sorts of nasty things on the mics.
Wait, what?
We're dropping all the hot goss on hot mics.
These mics, I could use the sensitivity in my session just a little bit ago, because you had to be like here, and if you got any closer, you were like blaring.
There's like 3 mics. When I turned this one off, it could pick me up, but
What? The skirt?
I feel like
No, it's a store. It's called Spaha, and all they sell is nerd clothes. That's where I got the red lifestyle dress. That's where I got my Aurora pants. They also I love that place. And it's all like stretchy fabric, so when you're like a fat mom like me, you fit everything.
None of you are against this being recorded, or
Nope.
Are we
Broadcast
ready?
to the
Are we
general
ready?
public.
I don't want anybody to witness it. There needs to be no evidence that I was here.
I'm supposed to do a 1-minute introduction to nerds.
okay, great.
And I have it on here.
Oh no, we have a sponsor talk now?
No. It's not. I'm not a sponsor.
I'll let you talk. You're one of my people, so I'm not going to argue with this. New England represent.
Is that the computer with the?
Yes.
I mean, I'll wait till everyone's here.
I will always let my fellow New Englanders represent.
I believe so.
Yeah, thank you.
Cough drops?
I'm good.
Test the Q&A mics.
Do you need help?
I'm just trying to
How many cytometers does it
There's
take to
one
plug
at
in
the
a
very
flash drive?
At the very front. Yeah.
Test,
I got a C and I got a A. I don't know what's
Oh
better.
my gosh, there's a thing in front of this. I can crisscross applesauce. Oh heck yeah.
This is just one slide. So.
See, I got my weapons out. Consent, humiliate, and non-panic. Oh my god, where did you get that adorable?
Hello, farmers.
Oh, it might have been in that one.
Well, if you go down here, you should see USB
So I just
drive.
have
Probably a
one of those
stash.
little Peruvian clay buckets.
Mm-hmm.
But then get a hair extraction. So this was like roughly the same size. It goes in my backpack and it goes from thermos to here.
That's perfect. That's much better than like drinking straight from burning thermos, which burns you. But I got this one specifically to fit in my little Tom bin because I have the big one that I take with me camping.
Great.
So I hear you on that.
Did,
But you have to put it in a cup.
were
otherwise
you
burn.
told last minute that this was a thing, or?
It was. I think I got the email Thursday.
Oh, you were volunteered sooner than we did.
I got
Oh,
my
right.
email Saturday night. It was like, hey, the person who's going to be giving the 1-minute presentation is attached to this. Thanks for your cooperation. I was like, I guess I'm doing that.
Well, it's a little. I mean, it's a little weird because this is like of interest to everyone, and this is a very specific closed session. It's of interest to me this session, but so maybe that's why Ken did it. I don't know.
Yeah, the inscrutable will of the master. I have no idea. I just go with it.
All I know is he's one of my people, so I'm not gonna argue.
They say jump and I jump, mostly.
New England psychometrists gotta stick together.
Yep.
Yep.
I say as a non-New I'm close enough.
You're close enough. You're like almost there.
I'm upstate.
We
You're
take
like
upstate New York.
barrier.
Yeah. I have a home.
You're on the other side of the Yankees wall, but you know we'll still keep you.
Yeah, I'm a Leafs fan.
You're what fan?
I'm not a Leafs fan. I was just
Okay.
trying to think of any team that might annoy Yankees fans.
I don't know anything about sports. Ball
Me
fan.
neither. I just like live hockey because the violence is fun.
I I used to pay very
Getting
close
your
attention to the
sports
Red Sox because I worked at Dana-Farber, and if you left at the wrong time, you didn't get home.
Yeah.
That's a good reason to watch sports.
I don't work there anymore, so I don't care.
We've got
It helps
2
that
minutes left.
I
We'll let
recognize
some
a lot
folks
of
roll
folks
in.
from the last
Find their way up here.
Yeah, it's really easy to get caught in conversation. You're like,
What?
You're gonna do great.
Huh?
You're gonna do great.
Oh yeah, yeah. I think you're more nervous than I am.
I'm getting all my wiggles out now, so when I stand up there, I can be coherent like a laser and give a presentation.
I went last year.
The worst part is once I get up there, I'll be mostly normal. It's just the before,
Normal for you or for everybody else?
Yeah, you might
for
have-
me.
Okay.
I'm honest about it.
If you have anything you're interested in talking about.
Oh.
Wait, did you buy?
Well, there's a
did you
lot
buy
of stuff
for
I
your
could talk
presentation?
about. Will it be
Just to say, I've got like 30 of those.
No, I can think of something.
Yeah.
I can think of something. I was just talking with
I meant to get more hot water. I just didn't.
people working together to like
I couldn't make it work.
get up and running
The cold
a
water was
pipeline, if you want to call it that.
I might just not be
Just
smart enough.
establishing what the expectations are.
No, I think we're doing that.
3 minutes.
The last one.
They match. Did you notice that? They kind of match.
Flow analysis
Oh wow.
was
Now that you've said it,
Yeah, I can't unsee that. I liked thinking it more Northern Lights.
meant the whole
Yes,
issue
there you go.
around people just drawing stuff, you know, and then when you try to
Is Northern Lights just a
that
two
into
laser aurora? Is that the
There's
only
one
difference?
blue, one
data.
red, the 2 laser settings.
It's weird, yep.
And that's
The 2
kind of
laser
when
setting now since they rolled
people
the
hear a
small
little bit
aurora
about
over to the.
Yes. Yeah, I as a
So what's the difference
wasn't
between the
built.
Northern Lights and just?
for like
How many
standardization.
lasers and built and stuff you
It
could
was
put in
built
it?
to identify things.
yeah.
Oh, so it's not expanded.
Yeah, it's just like I need this. All right, you
We have a 4 laser
It's the only thing
Aurora
on
that
here that
became
could
a 5
possibly be
laser
that,
Aurora
therefore
because
I
we
take it.
stole it
Yeah.
from one of our closing companies and it's old.
Yeah,
and that was fine, but it went from having UV, violet, blue, red, to the yellow, green in it to match everybody else. Northern Lights, it's pretty much red, blue, violet,
Don't just do stuff.
and never anything more than that.
It's
But you
just
can,
smaller?
it is smaller.
Help us all come here.
Ours also, the power light doesn't work, so you don't know if it's on or off because you can't see it. So you have to put your ear up to it and be like to hear heartbeat. So, to be honest though, I see no reason to ever purchase a Northern Lights. I just don't.
Well, because you have to build that expectation, right? And then the
People like what they like.
question is how you do that with
Watch
cut
and
off.
flaw.
Almost inevitably end up
I'm so
skewing
confused
to the
'cause-
realm of subjectivity. Is it
Sora
time?
Sora Chikanda y'all.
Very good. Hey, everybody. Thanks for coming. I think we have a great session for you today. Before I get into that, I've got Dan Mielkars here to give a quick 1-minute presentation on. It's nerds, right? Not I say neural skid, and I feel like I'm wrong.
I mean, I say neural skid too.
Okay,
Everyone
all right.
else says nerd, so.
So it's nerds or neural skid. Anyways, it's a regional chapter of the ABRF. And without further ado,
Hi, everyone. Like Steve said, I'm Dan Mielkars. I'm the president of Nerds, or Neural Skid. We've been given 1 minute to kind of promote our chapter, and it was attached to this session for some reason, so here I am. I mean, I'm a flow cytometrist, so maybe that's why. So if you live in this map, up in the upper right corner, and you're an ABRF member, or even if you're not and you just work in a core, you can be part of Nerds. We have a meeting every year. It's great. Last year we had like 250 people at the meeting, counting vendors and scientists. You could scan that QR code to get to our website. This year we're going to be in Worcester, Mass, October 14th to 16th at WPI. It's going to be a great program. We try to cover like kind of a broad range of core technologies and administration. So if you're involved in any of that, you're perfect to come to our meeting. That's basically the 1 minute Nerds promo. If any of you, like I said, if you live in these states or Canada, Eastern Canada, come to our meeting. It'll be fun. Okay, that's it.
Thank you very much, Tim.
Just close this. Uh-oh, lost the mouse.
Oh, that should be on the bingo card. Losing the mouse.
Oh, we should send the bingo card out.
No, but we're gonna give you guys bingo cards.
Weird. Can we just all tap it?
Yeah, I'm gonna just yank this.
Go for it. All right. Oh, fun. Could I grab you? Yeah,
The pointer disappeared somehow.
Yeah, I don't. Okay, all right. Thank you. Sure. Little of the old black magic. Love it. All right. Well, welcome, folks. I think I probably should mention the housekeeping notes. I think the biggest one is silence your phones. Everybody here is cool with you photographing any of the slides. I just asked, so I'm sure. And I believe this will be taped. I don't know if that's still a thing, but that is that. So, hi, I'm Steve. I was asked to chair this session and immediately started sweating because I know not very much about complex data analysis. So I went to my fine folks at the research group and said, who should I get to talk here? And they did me a solid one because we have a great panel of folks to talk to you today. And I think at that point I'll shut up. Which one of you wants to go first?
I don't
Alex.
think wants is the word here.
Nonsense.
Which one are you?
No.
This one? Love it. Take it away.
Thank you very much. Hello, everyone. Thank you so much for coming here today. My name is Alex Wendling. I'm coming from the Virginia Commonwealth University Flow Cytometry Shared Core Facility. And I'm going to talk about how our core started managing to transition folks away from their older conventional instruments and their basic 4-color panels and to start actually branching out and going into more high parameter spectral stuff. Because we received a Cytec Aurora finally after trying to get one for a number of years. And we got a lot of difficulty trying to convince labs to adapt and pick up this new technology that we had brought in. So in order to facilitate that and help get people to get that foot in the door and transition to more of a high spectral, high parameter workspace, we developed this in-house panel that we would then sell to our users to then perform some of their stainings as they're beginning to learn more about how they can do panel design, how they can look at some of their data sets, and then just transition into this new field, this developing field that we've got here in flow cytometry. A little bit of background about me. I've been with the VCU Flow Cytometry Core for the last 3 years. Before that, I was with the University of Virginia Flow Cytometry Core. And before that, I worked in one of the research labs at UVA where my manager used flow cytometry as a hammer and every problem he encountered was a nail. So I've been doing flow for the last 15 years pretty regularly on both sides as a researcher and as a core staff member. So as I mentioned, we were having a little bit of difficulty trying to get people to really adopt this new technology and move their panels off of their more familiar Fortessa or even their Canto instrument that they have been using for years. And so some of the issues that we found we were running into is just making that transition to some new unfamiliar technology. If we tell them you need to bring reference controls, they're like, well, what's that? Reference control just being the exact same thing we all know as your single stain control or compensation control. There's literally no difference. But right off the bat, they're hearing this new terminology. Then when you throw in stuff like cosine similarity and these new metrics that they need to take into account, as a biologist background, I know if you throw the word cosine into just a general thing, it's going to be, oh, this is more math heavy than I'm prepared for. This is a little bit more difficult than I'm ready for. And then we're not really beating any allegations when we got a metric literally called complexity score, which makes it so much harder because if you try to Google complexity score and figure out what it is, you're going to have a hard time figuring out how that calculation is done. Some folks in here might know that it is referred to as the condition number and stuff like that. But we've got these basic barriers to entry that make it a lot harder for people who are new to this field or have just more of a basic biology background and not a very strong flow background to migrate into this. And it makes that a little bit more difficult to transition. If I use the term eigenvector, I'm not sure how many people even here would necessarily know what I'd be referring to when it's just your reference control spectrum. And so just that terminology that we use makes things a little bit more difficult from the get-go. And some of the new and additional considerations, and even some of the old considerations that people don't put enough thought into, become much bigger in the spectral landscape. Things like spillover spreading, where you need to really factor in co-expression of markers on different cell populations, making sure that you're getting a good resolution between your single and double positive stains. Or, as we'll hear later from Laura, your autofluorescence considerations has a much, much bigger impact when you're on a spectral landscape, especially if you need to do autofluorescence extraction of multiple populations at once. That's not something that we would typically need or even could take into account for many conventional instruments. And then finally, one of our biggest hurdles has been just the cost of antibodies. It's so hard to convince a lab, hey, you need to buy 24 new colors for this panel that you want to do. That's going to be a $1,000 drop right there, and we haven't even started in optimization. Nobody's going to really want to do that unless they're a new lab coming in, swinging out like big startup funds. But we don't get too many of those necessarily time to time, and so we need to figure out ways to drop that cost and help bring them in so that they can do things more cheaply to figure out what they're going to do, especially because a lot of these beginning labs that aren't used to a very large environment are going to be doing more exploratory panels. They want something broad because they don't know what they're looking for. Nobody wants to buy an entire vial of antibody only to find out that they don't need this marker and they've got 90% of it left and they've dropped $400 on it. And then we also run into a general lack of understanding of some of the advancements that we've made. We get labs that are coming in and they're just like, well, my PI trained me to do this 4-color panel. There's postdoc, and that's how I've been trained, and that's how I'm supposed to train my next generation of students. And you get this kind of momentum built up by complacency, and those things are harder to kind of work out because you've got all of these multiple generations of people that are kind of backing up this, we can't make these transitions and stuff. And just the idea that we've been doing this and this is how it's worked. I've had two different lab members from the same lab come in. One of them is doing a T-cell project, the other one's doing a macrophage project. And it wasn't until like 2 months after assisting them, I realized they were staining the exact same mouse. One of them would handle the T-cell panel, the other would handle the macrophage, and then they were combining the data sets later. They were just splitting up the work, and they would come in and run at different times just because that's when they were available. And as we all probably know, that's going to have major impacts in sort of batch processing and effects from just sample handling. So by being able to combine those into a single workflow, it's going to make stuff more consistent. It's going to reduce the amount of sample that they need, and it's going to reduce their overhead time. So being able to provide something like this from the get-go just makes it a lot easier for new labs to want to pick it up. The way that we went about overcoming some of these obstacles is from a few different ways. We first started by just gauging VCU faculty interest. We sent out multiple surveys, we met with a lot of different groups to figure out exactly what populations people were most interested in. The panel that I go over today is in no way meant to be an OMIP or say, this is the type of panel that you should all implement at your place. This is just how we did it at our institution, because these are some of the factors that most of our folks are looking at. If you've got a much more heavy B-cell group at your institution, this probably isn't going to be the panel for you, and that's perfectly fine. This would be how you would go about setting up your own panel. Once we were able to pick out some major populations of interest, we then developed a 23-color panel that we would then distribute and sell and basically keep maintained inside the core facility for the faculty and staff. And when we were designing the panel, we made it so that a lot of the floors are very easy to substitute out and replace. So we managed to pair some of our markers and fluorophores with stuff that might not necessarily be of interest. If that's the case, they could easily drop it and substitute with something that would be of much more interest, which I'll get into a little bit later. We also prepare all of the reagents in-house, with the exception of the zombie near-infrared that we tell the users to get themselves, and we provide them with catalog numbers. But all of the remaining reagents, such as the antibody cocktail we provide brilliant stain buffer alongside that. That way we know that we don't have to rely on the users buying that themselves, saying that they put it in or just completely forgetting. And then we end up with a whole bunch of polymerized antibodies and aggregation forming. We provide that for them, and we don't have to deal with that kind of like troubleshooting later on. In addition, we provide FC block that we make in-house so that we've got a good idea that since these are mouse panels, that they're not going to have a wide amount of non-specific binding due to no FC block included, which we've found that a lot of our users will either forget or just neglect to add. And then as well, we've got a separate stain for any intracellular antibodies that they might be interested in, most commonly FOXP3, because we've got a lot of groups who are interested in T regulatory cells. When we provide that, we also give them all a step-by-step SOP that they can then follow along. It's got a lot of documented information on combined protocols for tissue sample prep and staining. So they've got everything from isolating their samples, staining, to when they bring them to the core. All of that is written for them that they can follow along, and any questions they have, they can reach out to us with any of those details. And that is a living document that we continue to update as we get new considerations for tissue type or sample handling. And then we provide templates that are already preset for data acquisition. We use SpectraFlow, we've got Aurora instrumentation, and data analysis on our FlowJo, as well as Cytoscribe software. So we can very quickly and easily get users to hit the ground running, and it helps us streamline both training on the instrument and data analysis. It gives them sort of a guided on-rails system to follow along with doing the data analysis and data acquisition, what they can expect, and where populations should generally be expected to fall. And then most importantly, once we're done with this, we try to move them away from this panel and do a more hypothesis-driven, focused experiment on their own sample. Something that's going to be much more appropriate for what they're looking at, generally because either their own personal autofluorescence in their tissue or sample types, or focusing on markers that are more specific to what they're looking at, instead of this more broad panel design. And also, we don't want to be known as that one core that only runs a single panel. So as I mentioned, we keep the panel fairly versatile. So as I said, it is not meant to be an OMIP. This is not going to be a collection of the best fluorophores to use. Even when the panel was originally designed, some other fluorophores have come out that could possibly be used to replace some of these with less spread over spilling. But the idea is that we've got pairings such as FOXP3 on Alexa Fluor 488. And for users that might not be interested in doing the intracellular, they could then drop that, and now they have both 488 and FIT-C fluorophores available, which are going to be more readily purchasable from many different distributors that can match some of the marker populations that might be more rare and available on their particular color of interest, rather than us having to play musical chairs and shuffle around the entire panel design because they want to do a single substitution and we can't find it sold anywhere else. Similarly with APC and KLRG1. And we've also got a list of other drop-in available floors, but we intentionally keep this panel limited to only 4 additions. We don't make this as necessarily a backbone panel for people to use, but rather something just to get their feet wet and into more of a high spectral space so that they can transition away later. And we utilize some of the projects that have come out from Peter Mage, Andrew Konechny, and Florian Mayer's unmixing dependent spillover spreading paper that has come out fairly recently. And so the general workflow whenever somebody comes in is we always start with a consultation. Users aren't allowed to purchase this without first going through a consultation where we can then discuss, most importantly, what reasonable expectations there are. Some people think that as soon as they do this, they'll get all of the data from all of their samples and everything will work out well the first time because it's a pre-built panel and no titrations or anything need to be done and everything will be hunky-dory from start to end. And we tend to, be upfront and say, this might not be the most appropriate for your panel, but here's some of the other populations and tissues and stuff that we've looked at. And we make sure that, if they've got a specific tissue type that we've never run or any other unexpected things, we can try to address them. If they're using doxorubicin or something in some of their stains, that we can anticipate that they're now gonna have an extra autofluorescent or an extra fluorescing dye in there or drug treatment and whatever else we might need to do in order to anticipate that. When we do the consultations, we provide them with the SOPs and all of the reagents afterwards based off of how many samples. Make sure that they've gone through and that they've got all the reagents that they'll need and that they're able to conduct all of the performances and they have everything in their lab. If not, we then pair them up with other facilities that might have Eppendorf spin down centrifuges and things like that, so that they'll be prepped and available to do any of these processes that they need. When they bring their samples to the lab, we have them bring unstained samples for any tissues that they might be using in any conditions so that we can properly match autofluorescence extraction. So we do give them that they'll need to do in addition. And that way we also build up a little bit of our own unstained and autofluorescence library that we can then pull from and use for our own examples and resources. After we do the data acquisition, we always do a follow-up consult, looking over their data with them to identify any potential issues such as unmixing, autofluorescence signal, whether we might need to substitute beads for cells, and so on and so forth. The acquisition templates and our unmixing templates that we have are already pre-recorded. So the acquisition templates we run with both slingshot beads and splenocytes that we run in the core on our own samples anytime we get a new bead lot, a new antibody lot, or if we get any sort of maintenance on the instrument, we'll redo that then, and then we'll have that updated reference controlled library that we can then pull from, which is beneficial not only to make sure that our users are gonna have more up-to-date controls from these samples, but also we can check for our own information how this panel might be changing on the instrument over time, making sure that our instrument's not having any slight drifts in anything, or some of our own antibodies aren't shifting unexpectedly from being pulled in and out of the fridge. And then this allows the users to kind of get started a little bit more quickly by just pulling in their samples. We've got the gates preset for them because a lot of users might not be familiar with, like, where do I position some of my gates to do this unmixing? What's considered positive? Is this what BUV 615 is supposed to look like? So on and so forth. And then, as I mentioned, we'll then follow up with these users again afterwards. The data analysis templates we have available on Flojo. We've got Flojo site licenses that we distribute out to various labs at the beginning of the year per request. And we also have data analysis computed with Flojo preset where users can then pull in some of the templates that have some basic gating strategies and everything set up for them, as well as exporting statistics and figures. We also have a built-in template with Cytoscribe, which is BioLegend's cloud-based data analysis platform. That one has the same pre-gated populations, but does not have the same, you can't import the same statistics and figure generation yet, but I have been told that it's on its way. And so once we have that available, we can drop some of the protocols and have those pre-generated stat lists already made for them. And then both analysis softwares have protocols for generating things like TSNEs and UMAP dimensionality reduction platforms so that they can generate their data quickly. And all of this, as I mentioned, helps keep everything on kind of a guide rails so that we've got users who are interested in not only performing the analysis and stuff, but want to be trained in the software. So this gives us a nice, easy kind of way to introduce Flojo to folks who can then see how their gating is done. We can recreate the gating alongside them, show how sample import is made, and then show kind of what examples of different populations should be. The general gating strategy that's provided from it allows us to look at a few different populations such as NK, NKT cells, CD8 and CD4 populations such as naive, effector memory, memory cells, T regulatory, as well as M1 and M2 macrophages, and some very basic B cell populations, but we have recommendations for labs who are interested in it, what's some available drop-in colors that they can use for more distinct population gating that they might be looking at. We've already run this panel across many different tissues. I've only got a few examples on this slide, including brain, blood, mammary gland. Brain, we're also able to look at microglia with some of these marker combinations, but we've run it on many other tissue types in addition to these throughout the different VCU faculty and labs that have come and brought samples in. A lot of the users have preferences in just terms of what populations they want to look at. And so for some of those, we can do things like split populations up into lymphocytes and myeloid populations. And we can cut down on kind of the amount of information that's in their face by showing t-SNE plots off of just T cell or monocyte gating so that they can kind of focus on just what they want to tell with their particular stories. And we've also done various panels alongside our tumor microenvironments, stuff like 401 T cells injected in mouse flank, high-MYC developing prostate cancer. We're able to look at those populations as well. We can see that there's some other populations of interest that our panel does not capture, and in those cases, we could use something like an EpCAM marker to then gate on other cells that would be within those. Our overall interest in the panel and our cytokine bookings has increased steadily and has been continuing to do so. Since we first introduced the panel in 2023, up through the end of 2025, 2026 is already looking to outpace pretty much every quarter in 2025 as well. So we've been having a lot of good interest with it and brought in a lot of new labs. 43 researchers across 29 different labs, both within the Massey Cancer Center at VCU and outside of it. So we're attracting not just cancer center labs, but also others such as our dental school, which has been kind of interesting. And we've distributed over 3,000 aliquots of our panel as of the end of 2026. And we are currently in the process of developing a basic human panel as well that we've been using in a couple labs with some pretty good success as well, but we haven't published any of that data yet. And speaking of publishing, another great feature that we found is that by distributing this, it's been a lot easier to keep track of which labs have been utilizing this panel, have had access to it, and making sure that they provide appropriate levels of attribution and recognizing the lab. Because on the SOPs that you hand them out, you can slap on every single page, remember to attribute your core along with exactly what you need them to type in. And then when they go for publications, you have all of your methodologies ready for them to go to just copy and paste directly in there as needed so that you've got all of that available. And it makes it a lot easier for both them and you to not have to hunt down every single lab and say, hey, we saw that this paper's in pre-publication and we couldn't help but notice that you hadn't mentioned us at all, despite figures 2 through 6 being directly flow plots. What's up with that? On the topic of acknowledgements, I'd just like to thank everybody who helped me to work on this project and has helped me come here to talk with you all today. My co-staff at the VCU Flow Cytometry Shared Resource Center, Dr. Rebecca Martin, our director, Madison Isbell, our manager, and Dr. Shinyan Pei, my co-flow course specialist, the VCU faculty who have helped provide various tissue types, acted as guinea pigs for titration and practicing, development of the panel, and given us data to work with and present, and the folks at BioLegend who helped with assistance in setting up the Cytoscribe templates and dealing with my great number of questions of how does this software work? It's confusing to me. Dr. Lisa Pinnell and Dr. Santosh Puda and Dr. Rebecca Nickel. Thank you all so much. Oh, that's
So I could actually start my entire deck saying, hey, what he said, because we've actually done a lot of the same steps, and you can sit there and design the most beautiful panel, and it's still going to have problems. So I'm Laura. I've been doing this for more years than I want to count. Before I was at AstraZeneca, I've been there for coming up on 11 years. I spent about 9 years running a core at Mass General for the Stem Cell Institute. And I have dealt with autofluorescence longer than I like to admit. So I would like to see a show of hands. Who here has never dealt with an autofluorescent problem in their panels? Lies. So even those of us that have been doing non-spectral cytometry have spent many years dealing with this. We all know if you're going to do color versus color, you know that you're going to see that diagonal line. Well, the cool thing about spectral cytometry is it gives you a lot more better ways to deal with this. I'm just going to tell a little bit about why it's a nightmare and how to try and wake yourself up from it. But before I go any further, I'm required to give the conflict of interest disclosure that yes, I am an employee of AstraZeneca and I have stock and stock options. All right. So those of you that read Shapiro know that one of the most important things in doing any flow cytometry experiment, besides there is no magic and you darn well better filter, is you really need to know your cells before you start going forward. Knowing thy cells is one of the most important parts of doing panel design and understanding autofluorescence. So I'm going to talk a little bit about common sources. I'm going to talk a little bit about how I use SpectraFlow for actually getting the problem a little bit out of the way, choosing the correct negative controls, and just a kind of side note about why we should never be using per CP Sci 5.5 again for as long as I live. All right, this is my favorite part. Bet you never thought you'd see this slide again. So, Krebs cycle. I realized that this has been sitting in the back of my mind for a very long time, and we can blame it for one of the biggest problems, because intrinsic autofluorescence is something that has been causing so much pain for all of us for so many years. We know that cell lines are going to glow violet. We know that when cells get activated, they're going to start showing up green. Well, that comes down to this stupid Krebs cycle that we thought we would never see after high school. When your cells are metabolically active, they're going to be turning on different metabolites in their system. If they're activated, they're going to be turning on NADH, NADPH, and then they're going to be turning on FAD when they're activated. And those are honest-to-goodness fluorescent molecules. What? Our cells are glowing and they're supposed to? Yes. It's an honest-to-goodness readout that we can use to identify what's going on with our cells. We also have phosphorins, which will also glow red. And if you're ever looking at granulocytes, you're going to find this really cool red color in them that's going to interfere with all of your BB605. And then you're going to wonder why your macrophages are positive for NKP46. Ask me how, I know. So these are things that just happen with our cells. There's not much we can do about it other than understand it and use that to make better choices when we're trying to get autofluorescence out of the way. That being said, we can also use it to understand our cells. There's several papers that have been out in the past 30 years talking about how you can actually use autofluorescence itself to identify different cell subpopulations. So these are just two that I really like. And I was able to find these curves out of a paper in, I don't remember now, there we go. Metabolites. There's an entire journal about nothing but metabolites. And you can see here the excitation emission curves for NADH and FAD. They totally overlap with FITC and BB421. It is what it is. This is one that caused me no end of headache at AZ. So at AstraZeneca, we work with a lot of different targets. We work with a lot of different disease states. And when I first was hired in, I was part of the small molecule immuno-oncology team. I hadn't really worked directly with small molecules. And then I learned very quickly that working directly with small molecules is kind of horrible, because every now and then you get something that either makes metabolic changes in your cells and then they glow ways you didn't expect, or the small molecule itself glows. There's actually precedent for this. So the history of Hertz 33342 is it was supposed to be an anti-parasitic. And they dosed rats with it, and the parasites glowed and died, and the rats kept glowing. So then they said, oh, we found this really cool dye, which glows up in DNA. We can work with that. Well, that's the first case that I found documented of a small molecule glowing. I have plenty more. We can talk about this for days. Doxorubicin is one that's often used for, it's a cancer drug. You can use it for inducing senescence. And in this case, there was a scientist that was trying to see if she was getting changes in efflux when she treated her MCF7 cell lines with doxorubicin or treating it with CDCF. CDCF is known to be fluorescent, and it's supposed to be upregulated and downregulated as you affect the transporters. So I was like, you know what? I'm going to run this on a standard cytometer, but I'm going to put this on the Aurora just for fun. And I got this curve. And I went, hey, look at this. I've been showing this to everybody in my group ever since. I'm like, Dox glows. And they're like, Dox doesn't glow. I'm like, look at that curve. That is the honest-to-goodness changes induced by doxorubicin. And it's not just metabolic changes in this case, because if you're inducing senescence, you're going to be expecting more of a change in the fluorescent regions. None of those. This is in the reds. This is the compound itself is glowing. So this is what it looks like if you're trying to interpret it non-spectrally, conventionally. This is me looking at the green emission versus the red emission. And it is a horrible smear. And I said, I wonder what happens if I try and do spectral unmixing with this. And oh my gosh, I can actually see them apart. So this was a case where I realized that maybe we could take these compounds, which independently glow. Look at that. Look at those rings. I love this picture. They're rings. They're generating fluorescence. This is expected behavior. And we can just treat them as new fluorochromes and let the software do its magic and unmix it. So we know that cells have metabolic activity that can glow. We know that sometimes our drugs can glow. We also know that some cell types are worse than others. So I just want to give all the props to Annabelle Kina at Boston University. She showed this slide at her at the Cytech user group meeting in Philly many years ago, and it has stuck with me. And this is an example I show to my scientists all the time. So we all know PBMCs are heterogeneous. You got myeloid cells, you got T cells, you got all the stuff in there. So you've got the things that are high and low autofluorescent. Cool, I can work with that. So this is looking at standard lung samples, and there's going to be a lot of alveolar macrophages, there's going to be a lot of other lymphocytes, there's going to be all sorts of heterogeneity in there, and you can see some of the cells are highly autofluorescent and some of the cells are not. Then you get a smoker. I love this slide because it means that you can't just rely on guessing where the autofluorescence is going to be. Knowing what happens when you have a person making poor health decisions and how it's actually going to be affecting their cells, you can see by the naked eye the effect the tar has had, and you can see by the spectral plots there what effect the tar has had. And, you know, it's going to kill the patient, but it's also going to kill your data. So when you're designing your panels, you really need to pay attention to what the effect is of the disease, of the compound, of the metabolic changes in your cells. So this is the summary on that one. What can cause autofluorescence? All these things. In reality, you don't care. You don't care what causes it unless you're going to try and chase it down. And I can tell you, it's a really fun railroad to go down because you can't get off it. You're going to be trying everything and researching things and what happens if I induce this and induce that to try and identify which autofluorescence deals with what specific aspect of your cells. When it comes to flow panels, you don't care where it comes from. I'm going to show you how to take it into account. So the first thing goes back to the first slide I showed you with the Krebs cycle, talking about the different places where we see common excitation and emission from our metabolites. And on the Aurora, what I found is this is excited on the UV in the like violet regions, excited in the violet on the violet regions, like 450 or so. And there's this weird little one that shows up on the violet laser in V12, which is kind of like in the 630 or so, which tends to show up in granulocytes only in humanized mice PBMCs. Weird. So I like looking at V12. Bravo 3, that's about the same places you're going to see Lexaflor 488 and 515. That's going to correlate with changes in FAD. I'm always going to check in the middle of the yellow-green. That's where I tend to see the porphyrins show up. And then I always check in the red. And that's because you always have that user who has a weird red signal. And you say, did you use phenol red? And they say, no. And I say, are you sure? And they go, well, I washed it out. And I'm like, well, you didn't wash it out because it's still there. So I always look there because you will find phenol red that tends to stay embedded in your apoptotic cells, and you can kind of use it as a way to get them out of the way. So take a look at those. Those are the common places that I found. If you're going to be working with compounds and other things, you're going to find other mysteries. But for general cells, you're going to find that they're going to excite and emit in these regions. So looking at them first is going to give you a lot of leeway and a lot of, well, I should say, a lot of steps forward into identifying the right pops. Okay, here we go. So that's when everybody starts going, I'm just going to draw a couple of plots. What's up, sir?
I'm so sorry to interrupt.
Go ahead.
Did you sort those B12 violet high positives?
No.
Factor was it positive?
No, because number one, we're in drug discovery, so we don't care. But number two, full disclosure for everybody in this room, my most complicated sorter is a Sony MA900. I mean, it's on the list of things I would love to pursue, but it hasn't been a priority for my projects. So, no, you're fine. So the next thing that happens is a lot of people say, well, I'm just going to draw a couple of gates and move on with my life. Well, the problem with autofluorescence is it's super duper heterogeneous. You can't just gate bimodally on 2 parameters and hope for the best. You need to gate on each population and examine it. So in this case, what you can see I've done, and I'm not even going to try with the laser point, I'm just going to point, is I've put yellow-green 7, which is where you're going to tend to see granulocytes, versus UV 7. So that's where I'm going to tend to see a lot of metabolic activity from NADH. And looking at that, it looks like I found 4 populations. That's fun. So what I do then is I look at each population on scatter, I look at each population on the spectral plot, and I look at each population against those other ones I showed you. So in this case, I happen to then look at one of those populations at green versus UV7 again, and I found inside that, it's not one population. I actually found 4 discrete populations inside there, because you're going to have a lot of changes going on across your population. So you need to look at all the different markers against each other to identify which ones are truly an independent population. So don't be afraid to gate too much. Be afraid to gate too little. You will miss it if you just do one big gate, and you will be stuck with heterogeneity with that, and it won't work. You need to go down and down and down and down. And don't worry about finding things that are the same 16 times, because that's when the modern software packages will save your butt. When I first started learning to do this about 5 or 6 years ago, there was no automated way to determine if your autofluorescent populations were the same or not. So you would gate on anything that you thought looked different, and then you would put them all back in as new fluorochromes, and then you would unmix, and then half of them would be the same. And then you'd have to sit there and be like, well, which one was the real one? Which one was a ghost? Which one? So you'd spend hours dealing with that. Well, most of the modern software packages, at least in Chorus and SpectroFlow, give you the ability to do all that drill down and find 20 different autofluorescent populations, and then the software will tell you which ones are actually unique. You don't have to sit there and chase it. It just says, hey, Laura, by the way, population 3 and population 7, they have a similarity in 1. You gated on the same thing twice. So what I've learned, if I have a lot of things come up with high similarity, it means I gated on the same population, and there was a lot of overlap when I did all that drill down, and then I can go and I can identify which ones are duplicates, so to speak. One of the major recommendations I have, though, is we all know that your single stain controls should be as bright or brighter than the population of interest. This also applies to autofluorescence. So what I'll do then is, in this case, I'll say, oh, well, it says that P9 and, what was it on here one? It's been a long time since I looked at this. It says that P7 and P3 are the same. Well, then I have to go figure out which one's the dimmer and which one's the brighter. And I'm going to delete the dimmer and keep the brighter. Because if you keep the dimmer, it's going to be incorrect when you actually move on to your extraction. So you find the ones that are similar, keep the brighter ones, and then you end up having the ones that actually are distinct. This is a pain in the butt. There's no way about it. With the standard way of integrating it into most software packages, as is today, it's work. And you need to make sure that you have the right controls to make it work, but it can be worth it. So this is a CD34 engrafted humanized mouse. So what that means is you have a mouse that's immunocompromised. you put some cord blood in there, and it ends up recapitulating an entire immature human immune system. It's got T cells, it's got granulocytes, it's got everything you need to get a really good grasp on how your drugs are actually affecting a competent immune system with a tumor-bearing setting. And the upper corner there is showing what happens if we look at human 45 versus mouse 45 and human 33 versus human CD3. And you can see with no autofluorescence extraction, I can see my 45s. I got that weird ghost population in the middle. I can see my CD3, but it's a mess. And then if you look at all the plots to the right, that's looking at fully unstained. That's just, I did unmixing based on single stain beads to find all my populations. It is a mess. You got your picture? You want the next one. That's what happens if you do your work. I'm very proud of that. So I'm going to show you again, before, after. That is the same two files. I spent all that time going through the unstained, identifying the populations that correlated with my metabolic differences, pulled them out, put them in as new controls. Here's another case, though, where actually identifying it and figuring out who they are can be helpful. So this is a case where I was working with just human blood, found this weird population, was like, that's cute. Turns out it correlated with my granulocytes. So when I did my autofluorescence extraction of this one, I had autofluorescence debris, autofluorescence this, autofluorescence granulocytes, and I actually, it's not shown here, but I wrote it in the file in the end, AFO2 grams. And we were able to use that as a surrogate for CD66B because we hadn't put it in this panel yet to identify granulocytes. So you can actually, if you do your homework and identify the populations, use it as a marker to identify your cells. When you're designing panels, you really need to pay attention to this. And you need to pay attention to each one of those different subpops that you found. And you can use that to guide you on what to put what color where. So in this case, you can see your granulocytes do have a lot more emission in the red channels than your lymphocytes. So in this case here, what I might choose to do is the places where I see a lot more autofluorescence in my grands and a lot less autofluorescence in my T cells, I'm probably going to put my T cell markers in there. Because if my granulocytes are in there, it's fine. I can deal with the unmixing, and I've got more signal-to-noise window. But I don't want to go putting my granulocyte markers in places where the granulocytes are already autofluorescing, right? Because that just gives a lot more gaps, a lot more clarity. It's not as easy to see that with these PBMCs, but it's totally easy to see if we go back to those lung samples. So in this case, I'll never forget Anna pointing out mind the gap. So you can see with the lymphocytes, you could put your markers anywhere in that spectra, and you're probably going to get great signal to noise. It's going to be fine. If you look at typical healthy alveolar macrophages, I'm probably not going to put anything in the middle of the violet. Probably not going to put anything in the middle of the UV, but I could put tons of stuff in the red, the infrared, and get really clean signals. You look at that smoker, there are teeny tiny gaps in there. And the rest of her talk was actually about how she chose only those gaps where she was going to put the markers specifically for her diseased alveolar macrophages. That's where she had to put them. Because if she put it in places where there was high autofluorescence and off-scale autofluorescence, you would never see that signal. So you want to take your healthy cells, you want to take your diseased cells, you want to take them unstained up front, and you want to look for things like that. Because that's going to give you a better idea when you go to design your panel of where to put markers. If there's high autofluorescence, don't put your markers there. If there's low autofluorescence, good place for your markers. Do it cell type dependent, and it'll be prettier at the end. Also pointing out here, this is not a bad t-SNE. This is Anna ended up taking the cells that correlated with the disease state and with the non-disease state. She ran it in omic through OPSNI, and for those of you that don't know, she actually wrote OPSNI, so she's a goddess. And she used this to find all these different subpopulations which correlated with unique autofluorescence, and you can do that as well. You can put it through any dimensionality reduction, you can put it through any clustering, and you can find those subpopulations, and you can go back and forth. And especially with FlowJo and Hyperfinder, you can potentially do that and find different gating populations which you could use on a FACS Discover platform. I don't have one, I can't do it yet. But it's something that we're looking into doing as we're moving on further with analyzing data on the A8 and reinterpreting that autofluorescence when we get our S8 next year. Don't tell David I said next year. Okay. And back to autofluorescence and chorus. Yes, chorus can do it. Chorus can absolutely do it. I can't do it yet because I'm not good at chorus, but one of the members of my team, Jianpeng, has been working on a lot of autofluorescence extraction in there. And she says it has a lot of the other same abilities of gating and looking for similarity and moving on. So if you're going to remember anything, this is like the major take home. I have a couple more slides after this, but you always want single cells. You always want single beads. You always want unstained cells. You want unstained cells for every tissue. You want unstained cells for every treatment. You want unstained cells for every single thing. Because if you don't find it now, and you don't have that control, then your users are going to say, what is this new novel population? And it turns out that it's an autofluorescent population. And no, your B cells did not start turning green, they got activated. The other thing that I found that I'll do, at least for singles, to make sure that I can get better signal to noise, is I'll mix everything when it comes to my single stains, but I don't mix my unstained. I will never mix my unstained, because that removes the ability to identify who is the problem. And then I will often start by unmixing with beads, because beads behave. With cells, it's a little bit more of a problem. I'm going to show that in a second. And you can also try different capture particles. And I want to thank David for posting about capture body, because I really want to play with those. So it's a new bispecific molecule where you have IgG on one side and is it
45.
Yeah, 45 on the other. And so you can kind of make a surrogate on real cells of new colors. And it hasn't been published yet, but when that's published, I'm going to play with it. So the last one is when you're doing cells for your controls, everybody just goes, I'm going to put my unstained cells in as the control. No, you cannot do that. So different cells have different autofluorescence. What I'm showing you here is, again, PBMCs. I've gated on the ones that are positive for live dead here, just to kind of show that even among the live dead, there's different populations. Actually, I'm sorry, that's not live dead. Don't listen to me. That's autofluorescence, but I labeled it as live dead because I was troubleshooting somebody's experiment. I did this like day of, and I spent literally an hour trying to take this experiment apart. And they were using full unstained with all of the different subpopulations as their unmixing control. So if you look here on the y-axis, you can see that UV7 is on here, and it's correlating with the different autofluorescent populations. And you can see here, here's their CD8, and their CD8 is correlating with the population that's lower. Here's their CD11B. Their CD11B is correlating with the population that's higher. What that means is if they use a heterogeneous unstained as their negative control, it's going to unmix incorrectly for both. Just like if you have your compensation control, it's going to overdo it or underdo it. Same thing's going to happen with your unmixing control. If you don't have the same autofluorescence for your positive and negative, it's going to unmix incorrectly. You can't do that. And beads will do it too. Every now and then I'll get a bottle of ultras and they'll just have two populations in there based on autofluorescence. And you'll always have your unmixing come out totally wrong. If you look at this one, you can see that it's a bottle of unstained ultra beads, and it has two populations, and only one of those populations is staining for your antibody. So this is why I will never, ever, 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 ever just use the unstained peak for unmixing, even with beads. I will always use a fully unstained bead droplet, because if you have this problem where the beads start to degrade and there's heterogeneity in there, you can take care of it. So in this specific case that I've shown here, what I did in the end is I gated on this population at the top. I used it as my universal negative for my beads, and then I just gated on my positive for each of the single stains. And so I was able to get that autofluorescent problem out of the way. But I had to have the fully unstained to identify it. So with cells, you need your fully unstained. With beads, you need your fully unstained. Did I mention you need your fully unstained? Did I mention I have this argument with my scientists probably on a daily basis? And then it bites them in the butt, and then they listen. The last one is to the humanized panel. So I showed this poster at Cyto last year. It was our CD34 engrafted humanized mouse panel. It's based on OMIP 69. And similar to how with OMIP 69, they went and they changed a bunch of colors around, we've been doing the same with our own. And one of the things that's been really nice is we're able to not just move to more of the stable non-tandems, which tend to not degrade, We're also getting more and more opportunities to move some of our markers away from the autofluorescent spots and into the places that are not as much of a problem. So I wasn't able to put in a lot more of the changes in here, unfortunately, but we're also moving to getting more things in like the new BB818 away from like 510. So an example for this one, we still have CD15 and BB605 for historical reasons. We're hoping to put that somewhere in the red or the infrared, because that way when we have those myeloid cells, we're not going to have the autofluorescence interfering them as much as they are right now. So the new modern fluorochromes give you a lot more flexibility in getting your autofluorescent populations in colors that are less of a trouble. When you're looking at the raw similarity, it's still going to be a pain in the butt, but you're still going to need your controls even with these new colors. Don't skip. So overall, autofluorescence, it sucks. Sorry. There's not much we can do about it other than use it, look at it, and build our panels around it. Always, 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 every single unstained, every treatment, every tissue, identify the autofluorescence, extract it out, then design your panel, and you will be much happier. So I just want to thank all these people whose data I stole for this talk with permission. So yeah, and thank you guys. Oh, and also I have to say that every single name on this slide is also an employee of AstraZeneca and owns stock or stock options. And special thank you to Annabelle Kina, who is not an employee of AstraZeneca. She runs the core at Boston University and taught me a lot about what I know. So thank you guys.
So hello everyone, my name is David Ruck. I'm at the University of Maryland Greenbaum Comprehensive Cancer Center in Baltimore, Maryland. I work with the flow cytometry steroid resources. And following up on what Alex and Laura have talked about, I have titled my presentation, No Cells Left Behind, Wrangling the Full Biological Discovery Potential Out of Spectral Flow Cytometry Datasets. So as many of us are aware, spectral flow cytometry is awesome. We're able to resolve significantly more markers than we could with conventional flow cytometry. And because of the fast acquisition speed, we can acquire far more events per FCS file than we ever could with a mass cytometer. Consequently, we have analytical breadth and depth that we didn't have previously that we can use to characterize rare cell populations that in other days we would have had to sort with a cell sorter before we were able to look at them. In the process of acquiring these very, very rare cell populations, oftentimes we're acquiring millions of non-target cells that still have pretty extensive phenotypic profiling within those same FCS files. And in the context of human clinical data, where your human clinical samples are of limited biospecimen, this can be particularly invaluable. Now, if you're only interested in a single cell population, your analysis process is not going to dramatically change. You may have a few additional gates, but you can still do what you were doing for conventional or mass cytometry in that context. However, if you're acquiring 3 million cells to only look at 100, and you're interested in what's happening to all those other cell populations within these rare clinical samples, the breadth and the depth of the actual data we're acquiring can complicate the analysis. So let's step back and let's think about something in the context of a single marker. If we split all the cells by that marker into either positive or negative populations, and then we repeat the process for all the markers within our spectral flow cytometry panel, at the very end, if we think about it as grouping cells that all share the expression of the exact same markers, we can end up with stuff that looks kind of like a cell cluster. Now, if we think about how this process goes out, it's basically combinatorial and it's exponential, and we can kind of estimate what would the maximum number of these clusters be at the end by just taking 2, raising it to n, with n being the number of markers in our panel. So, if we think in context of other days when we had a 9-color conventional flow cytometry panel, At most, we would only have 512 of these clusters present, which, as my colleague David from Michigan put yesterday, graduate students are cheap and desperate, and they could definitely get that done over the course of a weekend. However, if we're thinking in the context of a 40-color spectral flow cytometry panel, that combinatorial adds up quickly, and we have over 1 trillion potential combinations at the end in just a single FCS file, which all the grad students in the known universe for all their lifetimes would not be able to manually gate all those differential combinations at that stage. So if we're thinking about it, regardless of what we're doing with spectral flow cytometry data, if our goal is to profile widely within these samples we're acquiring, we will need to at some point use either a semi-supervised or a unsupervised analytical approach to try to get an understanding of all these potential combinations happening in these other situations. However, spectral flow cytometry's immense potential when it comes to biological discovery comes at the cost of a trade-off. Namely, the quality of our final data depends on our ability to be able to infer the fluorophores that are present on these fully stained cells. How closely the fluorescence signatures in our mixing controls, both single colors and unstained, end up matching these same spectral signatures that are found on the full stained sample will dictate that final quality when it comes to the analysis process. So when this doesn't happen, you end up with unmixing errors. Unmixing errors can then cause batch effects. These batch effects can then lead to unsupervised algorithms going wrong. And this is how you end up in situations where papers are published claiming to discover a brand new biologically revolutionary cell subset that turns out to just be a very bad mixing error in a badly designed panel. So it's very important when we're working, and especially if we want to do more of these unsupervised data approaches, to always try to reduce as much as possible the unmixing errors that we see in our panels so that we don't have as much trouble downstream. So despite the very critical importance that our unmixing controls, both single colors and unstained, play in this process, we have surprisingly few tools that permit us to kind of preemptively screen our reference controls to see if there's any issues with them before we actually have unmixed and are now looking at full stained sample data. Our existing quality control algorithms, things like PicoQC, FlowAI, FlowCut, they primarily focus on laser or fluidic related issues. They're not characterizing the signature of our tandems. They're not looking at unstained autofluorescence. So unmixing errors can come from any number of sources, including tandems starting to degrade, additional autofluorescences popping up, as well as undiagnosed instrumental issues that maybe the QC for the particular instruments has not yet flagged. Any of these leading to unmixing errors can lead to batch effects, which won't let you be able to proceed for these unsupervised analysis, and it's going to take your person doing the data analysis significant amount of time to try to manually correct things so that they are able to proceed. So, for the last 2 years at the University of Maryland, we have been working on developing open source tool sets specifically aimed at addressing spectral flow cytometry's quirks. We are developing, using the open source programming language R, individual R packages that target these specific pain points that we have all been seeing over the course of today. These 3 R packages are nearing completion. They should be completely out over the course of the next year, and they represent 3 main focuses for our institution. The Luciérnaga package does spectral signature characterization. We have the Cytometry QC package that allows you to implement automated instrument quality control dashboards that you are able to interact and see how your instruments are changing. And then we started to touch on the semi-supervised analysis using the Coerba package, which I'll mention as we go throughout today. So focusing in on what Luciérnaga does, the way it works is if you think of what the current algorithms are doing, or what a current unmixing process is, we gate on a big population of cells, we take the average measurement across detectors, that value gets normalized, and that's how we end up with a normalized fluorescence signature. What I am doing in the context of this package is slightly different. I am normalizing the signature at the level of the individual cells. At that stage, I can then match cells that have peaks in the same kind of locations of relatively similar heights, and then from those raw FCS files that make up both our unstained and our single color controls, isolate out the purified, normalized signatures that I can then use to evaluate how things look. So in the context of if a fluorophore vial is in very good condition, there's no breakdown happening in any form, we end up retrieving very few signatures across long periods of time as experiments get processed. This is an example for BUV496. It was stable across the entire experiment run over 6 months, not a single issue with that antibody vial. By contrast, when the tandem fluorophore starts to break down, we start seeing the byproducts of this breakdown because those byproducts still have an antibody that still binds to the level of the individual cell and still contributes a little bit to the overall signature. So my favorite example is this APC Fire 810 single color that was being processed. It will normally have on the Cytek Aurora a signal on the R8 detector, but you can see there's a gradient of expression happening right around the R1 that we're picking up because we're measuring the signal across individual cells and just aggregating them together. Now, I've seen a lot of these examples that are because a vial has gone bad, but in this particular case, this was a vial that was performing perfectly well on specific days, but on other days, we're getting back this highly degraded byproducts. So whatever was happening during the experimental process, reactive oxygen species were being generated, which were damaging the tandem within the single color controls before they were shown up and acquired. And one of my projects over the last winter at the core has been setting up automation that basically, for every single SpectraFlow experiment acquired by every single user, we process the reference controls, we process the unstained controls, so that we are aware when this is happening for any of the reference controls in our facility before our user shows up with bad data 6 months from now and is wondering what is going on. This same process, this same approach of characterizing the signatures at the level of the individual cell is quite useful when it comes to looking at autofluorescence signatures. So as Laura mentioned, there's a lot of variation when it comes to autofluorescence. We treat it like a single color. Oftentimes it is not. It's very heterogeneous. But because the same approach allows us to sort things that have similar peaks of similar heights, we can oftentimes go in and quickly isolate out those particular signatures. And I have it set up so just spit them out as purified FCS files that are perfectly compatible with the current unmixing software. And instead of having to go to the effort of isolate out these individual autofluorescence tags, you can just generate it from your unstains and then still go through the process of making sure you're not just getting on debris, you're finding the brightest signature, and then plug and play to be able to get a better result. And unlike the current approaches with Autofluorescence Explorer or those TISNI dimensionality visualization ones that oftentimes are sorting cells that have similar level of brightness, but maybe still differences in signature for the second or the third peak, their signatures are matched, but there's just a variation of brightness on those individual cells, but they all retain a similar identification. This has proved quite interesting because when I first started my PhD, I had a very important question for my postdoc. I was like, what's the difference if I gate here on the positive versus 1 millimeter over on the positive? And I did not have a good explanation at that point. But one thing we found is quite interesting is like if you look at a very bright fluorophore that just has a moderate density antigen, and then you just sequentially bin across where you would normally gate, where you normally would be gating using the best practices that we know, and you're just retrieving what signatures are present on the individual cells within those bins. You can see for this APC Fire 810, all the way from like the 20th percentile to the 100th percentile, all those cells are coming back pretty much identical signature. There's very little variation. It's only when you start getting to those dim cells that you start having little bits of autofluorescence peaks start popping up on where you would expect it, the UV7, the violet 7, the blue 3. By contrast, we found a situation that the guidelines for good unmixing controls do not actually cover directly. And this is in a situation where you have a population that has a moderate cell and moderate density antigen, but is present on a dim fluorophore. So in this case, this is an example of the BUV615, which was on a CCR4 antigen. It was discernible by our cell reference control. It was gating, we could see it, it looked like what we were expecting. But when you're checking the difference of the signature of the cells based on where we're gating on the bins, there was a very large residual autofluorescence contribution present for the entirety of the positive. There was no way we could just gate on the very brightest cells without still ending up with a little bit of contribution of the signature. So if we had followed the best practices of grab at the average and then up from the top, you would have ended up with an average signature that looks like this. And this component here and that component there are still just little bits of autofluorescence stuck in your single color cell control. And these still end up impacting on mixing, but it wouldn't have been something we would have ever realized. So it wasn't until I took those specific fluorophores that we had identified and swapped them from cell controls to beads that we had a significant improvement in our panel, because these little bits of autofluorescence add to collinearity, add to the mixing dependent spread. So if we're able to identify them and able to remove them, that also significantly reduces the issues we have in our mixing. So all this approach as far as signature characterization is dependent on a very key assumption that our instruments are performing stably over time, that any reference controls I acquired today are compatible with those acquired 2 months ago, those that are acquired 2 months from now. So at our core, we have instituted a daily process dating back to at least 7 years now. Before QC, we acquire 5,000 QC beads, and then after the QC has happened, we acquire an additional 5,000 QC beads just to be able to see, has there been any difference by the QC software when it's adjusting the gains. In the process, when I joined the core, I set up an automated process to take these QC bead FCS files, process them to get the gain information, the MFI of the beads, as well as the percentage RCV, basically the spread of the beads that we're actually interested in. And with a little work, I've set it up at this point where we have a little dashboard for our core for every single instrument there. It automatically updates around 11 a.m. every day. So if our users are worried or interested in what's going on with our instruments, a few of them are. I'm still working on the others. They can just go to the dashboard and actually look at the Levy-Jennings plots and see how are things changing. But for those who are working within a core facility environment, the real advantage of this is we have early warning as far as lasers starting to fail. So this is a typical situation we see when our lasers are failing on the Aurora. We'll have the slow and gradual change in gain going on for different lasers. And we're, at this point, able to detect when this is happening about a month out from when SpectraFlow's QC would start actually showing fail marks. So we're able to schedule with our preventive maintenance, with our field service engineers, get them out before at any point the data of our users is being affected. And then same thing, about 2 years ago when the lot 2006 beads were rolled out, we were like within the same day having unmixing issues. And what we realized was the QC bead lot for 2006 was being damaged by bleach, which was causing these spikes in RCV, which if we were not like actively monitoring it, wouldn't have been flagged because it wasn't the UV tree, violet tree detectors, et cetera, that were being flagged. And the long story short of that one is we looked into it compared to before and after. Basically, the bleach is stripping an APC fire like a 10 molecule because that molecule is no longer present on the surface of the beads. It has a lower than a target MFI. The instrument assumes there's been an instrumental change, maxes it up until it hits that target. All the while, the instrument was fine. It didn't need that increase in gain, but now all of a sudden it's way higher than it should be, which is why the unmixing issues resume. Now, I liked bashing on 2006 because at that time I thought that was the only bad lot we had. But because we had retrospective data for the last 7 years, I did go back and check, and I have been able to identify other instances for the other lots where there was likely some kind of bleach-related damage. But at that point, we wouldn't know. The user would have shown up, done their experiment, had bad data, blamed themselves or blamed some other factor, and try to repeat at that point. Now, if it was just a regular PhD experiment, that might not be particularly impactful. But if you think of a clinical sample that was a one and done kind of deal, that can be particularly harmful. So something we're currently working on is we have the process of identifying them, but is there a way we can preemptively give us warning? Because in a core environment, we are busy. There's like half a dozen things burning down at any point in time. So is there a way we can automate an email to us saying, hey, please rerun this QC beads? They did not seem to be matching what is supposed to be going on. And then in the interest of time, I won't go too far into the Coerba package. Basically, if you think of the unsupervised algorithms that we currently work with, things for like normalization, checking for quality control, checking for clustering, dimensionality reduction, many of these are like black boxes. We don't know exactly how they work. We don't know if our t-SNE is actually properly gating things as we would do in the context of a manual gated specimen. So one thing we're currently tinkering with is basically if you implement semi-supervised analysis by basically going through an automated gating fashion, breaking everything into positive and negative, but basically encode that information with the individual cell, you can pass it through all these algorithms, see where it ends up, and then be able to fact check, this is what I would have done with my manual gates, how did it end up affecting on this particular one? And we've recently published a paper on cord blood innate like T cells where we demonstrated this process where we can gate on particular sections of the different TISNE PacMap islands, actually get back the individuals, rederive the frequency and marker expressions based on the manual gates where they ended up. So it's not full as the be-all as far as the analysis goes, but it's pretty cool as far as what we have. So the current status of all this work, all these packages are available via our GitHub. I'm currently fixing the bugs and updating the documentation, and I'm planning on submitting them to the bioconductor repository this year. But one thing I've become increasingly aware of over the last couple years is what good is all these tools if the community as a whole is not able to use them? So I struggled when I first learned how to use R. I got tossed into the situation during the pandemic, and there was no information because University of Maryland wasn't a computational cytometry expertise location. A lot of what I ended up figuring out was reverse engineering the BD at the time FlowJo plugins to try to figure out what was going on for the code. So one initiative we've been working on since February is our free weekly Cytometry in R course, which we're offering both in person and via YouTube live streams. It is aimed specifically at the target audience here, namely those who have prior cytometry experience but have no or very little coding experience. We started in February. We are going through different topics, building from the very basics, slowly through the intermediate and advanced topics. You'll eventually know how to do unsupervised dimensionality reductions. And we have had a lot of global interest. We have had over 2,000 people fill out the interest form. And currently we're averaging between 300 and 500 cytometrists who are tuning in to listen to me talk about a soapbox of interest for a particular week. So in conclusion, I believe spectral flow cytometry is the future. It has the immense capacity to really be able to characterize the human immune system at a depth that we have not believed. But we need to be able to figure out how to handle these quirks and how to handle this dimensionality so that we're actually able to create the algorithms that will help us be able to do these analyses in the future. And with that, I would like to thank everybody at the Flow Cytometry Core, including Dr. Fan, Dr. Rajan, and Michaela Trainer. I would like to thank my PhD supervisors, Dr. Christiana Cairo and Dr. Kristen Leik, my molecular microbiology PhD programs coordinators, Daniela Franco from the PIN program, as well as both the Cytometry Discord, the Reddit Flow Cytometry, and the Bioconductor Zulip, who have collectively over the last 2 years put a lot of ideas in my head that then turn into rabbit holes, which part of the effort you're now seeing today. So they get some of the credit and some of the blame as far as like, why am I still doing my PhD? And with that, if you want any of the resources, if you want these slides, they're all available on GitHub. All my information is available. So QR code away.
I think we have some time for
No,
questions. No, not really.
we have
but-
negative time.
Well, if you feel like you have time for questions, we can ask questions. Otherwise, we can end the session. I'm not going to speak for you guys if you got places to be. But thank you so much. Every one of those presentations was absolutely impressive. You have risen to the task, and I am absolutely blown away. So from the bottom of my heart, thank you.
Thank you for inviting us.
All three great talks, each with a piece. I'm going to jump on the autofluorescence because that's always been my favorite.
Oh dear.
I'm curious, I've read some papers where, like, with microglia, they've measured, where you can actually have measurable autofluorescence increases across those channels you spoke of. They could potentially make that a metric
Yep.
of aging. Has anybody that you know used that autofluorescent measure as a metric, for human or mouse type cells as opposed to, we know the plant people do it.
So I haven't seen it necessarily for aging, but what I will tell you is Shapiro specifically calls it out in page 292 of, and I'm not joking.
Page number.
I have the page, I have this memorized. Howard Shapiro put in Practical Flow Cytometry, 4th edition, page 292, that isonothyl fluorescence was used at cytometry, in cytometers, this is 15 years ago, as a cancer indicator. Like, this has been done. None of this is new. The only difference now is that instead of it just knowing with specific cell types we're going to see shifts, we know that activated T cells are going to turn green and violet. We know this. It's we're able to extract it, get it away from our fluorescence, and see if we can better identify it. Because all we've done historically has been like, okay, we see it, we know it might correlate with X or Y. This gives us a chance to capture it and measure it. I know there's a lot of papers coming out with that. There's been like the 2 that I showed in Cytometry A, and I didn't even realize that this is something that's actually been used in imaging. So if I talk to my imaging corps people, they're like, well, of course you can do that. And I'm like, well, thanks, guys. It would have been nice to know that up front. But I don't know of anything published right now that's talking at least with aging.
Is it true that, because I don't have a spectral cytometer, unfortunately, but is it true that you can parameterize autofluorescence, and then is that a sortable feature? Potentially, yeah.
It is. You can do it on the S8, which is BD spectral sorter. There's somebody there that might be able to give more feedback on that. We've done some work with that on one of my sites. I just haven't supported that, so it's not my jam. And then the Aurora CS will also do that sort of sorting. We also have, through the Discord, there's somebody we know that spends a lot of time on the Sony platform. So like there are 3 spectral platforms out here now that can go and sort these populations out. I personally just have 0 experience with them.
You don't have to have spectrum, you can come up with this combination of gates.
Yeah, no, that makes sense too. It's just the spectral is so elegant, and to be able to isolate a specific channel that has just a semi-elevated one, it might not be as precise on a traditional cytometer. I thought that was really cool.
What do you use Hyperfinder?
Say
Are
again?
you familiar with Hyperfinder?
No.
So BD has that, which came out of like 200 gig of posts, but like you can go to the find UD population, and it will tell you the grading strategy.
Oh, cool. Yeah. Actually, I think I didn't know the name, but yeah, I've heard of the
Yeah.
process.
And that's actually something that we've been working on with our A8 to S8 pipeline, is because if you want to do all of that dimensionality reduction, you can't do that natively in Chorus at this point. But what you can do is you can take the A8, you can do all your analysis, you can find these populations, you can use Hyperfinder to identify them, and then you can go back to the S8 and you can use that to guide your gating strategy to find those different populations. And then we can go down the entire imaging cytometry label-free discussion if you want to, because like that's our jam. But like that's the thing is it sounds like it's new. It's not new. Just now we have new tools to play with it.
May I follow up with one other question for the first gentleman? And this is just general logistics. You've had a panel that's matured, and you're now, I'm guessing, procuring all of the antibodies and having them run out. How has the stability of the panel sustained itself through various lot generations for the different markers?
For the most part, they've been fairly stable. We'll come up across like an oddball every so often. But yeah, anytime a new lot comes out, we rerun everything just because it's easier to set it up that way for us, really. So we might get a new lot of like APC, and then we'll just rerun the whole panel because we'll just have it. And that way we can just check and make sure that everything's being updated. So we're actually rerunning just single stains on mouse splenocyte blood and slingshot beads pretty frequently, but it's not a whole lot of time on our part. Our director has her own research lab, so they've got a large mouse group, and they'll just sack a mouse for us, hand us the samples, we'll do the staining, and we'll be done in a couple of hours. And then we've got a whole updated library that we just pop right back in, and it's been pretty stable. We've had some changes, and we determined that was usually an instrument performance thing. Then we get a PM done, and then we have to rerun the whole panel all over again. But for the most part, it's been really good, but we do get odd mouse samples, I was about to say patient samples, mouse samples, that are just like, we don't know what to do with these. We've had some brain tissue that's like, yeah, we've got all of these splenocytes stained. We need you to stain all of your brain now because we can't necessarily figure out what's going on. So we do have users then have to come back, sack a mouse just to do this single stain control staining that sometimes we're like, okay, we only need one of these colors, and now everything looks much better and kind of what we were actually expecting. But usually once they're kind of committed to the data set, they're more willing to do that. So it's easy to make that jump then.
Any other questions? Excellent. Well, thank you everyone for sitting in on the session. Now go party hard tonight and enjoy tomorrow. It's the last day.